this fancy website here where they're trying to build not just a new data standard.
Thanks for telling me, Eric.
Compatible with sort of like all the typical activity, but it's rather like a bigger system. So they have like a system for data storage, because oftentimes these data sets now get so big that you can't even access all of it, even if you had one data standard. They're building APIs so that people can use their analyses. So like you can have one data set like spike recordings, and then you can run like some 6 different spike sorters on top of it and actually compare, because they all use the same API. And then a specification language, so it's such that the whole thing becomes extensible. So how do you formally define a new data standard? Say that you have a completely new kind of a data set. How do you store your things? Of course, these things all exist in different labs, but they're not really all that compatible between different labs. And so they've partnered, I think, they have like, is it a Kavli grant, I think, that gives labs money when they accept this standard. By now, there's actually quite a sizable list of labs. That's a picture that I took here. So you can see there's quite some big labs on this list who've like adopted that NWB standard. You'll also note from that list that it's a very American list. That's one of the things I asked them about. I think it's just going to be like more outreach that is necessary, particularly through the INCF, the International Neuroscience Coordination Facility, which I know a lot because, of course, they have an anchor in Stockholm. So I'm actually thinking of pushing that a little bit when I'm over in Stockholm with some of the experimentalists that I know, if they don't want some free money for adopting a new data standard. Then there was at the conference, I'm not going to talk about the conference too much because I'm mostly going to talk about the GridCell workshop, but I want to talk about a couple of the posters and about this double-blind review experiments that they did with abstract submissions to COSYNE, which was quite fascinating. So they looked at review correlations, for example. So how reliable are reviews? There's clearly a correlation you can see here, but there's obviously also some studies that like might get a 7 or an 8 by some reviewers, but then a 3 or a 4 by the other reviewer. So the total correlation between reviewer scores is only 0.25, which I thought was kind of fascinating. Then there was this cool double-blind study that they did, where they blinded the reviewers to the change that you get from blinding a particular aspect. So sadly, but not all that surprisingly, the mean scoring goes a little bit up when you blind reviewers to the gender of the first author. That result is not significant, but it does show up in the mean, as you can see. What is also quite interesting, that the last author, when the institution rank is so like a middling university, it makes a big difference whether the authors are blinded as to the level of that institution. For like the top institutions, blinding or not blinding makes no difference, neither for the lower ones, but there's a rather dramatic jump in the score when you blind people towards the institutional rank of the last author. So that's all a little bit sad, but I think it's also really cool. And I think it was like the study was easy enough to do that I think every conference should do that.
If I remember correctly, the way they sorry if you said this, I don't think you did though. Mm-hmm. The way they did these reviews, it was either 2 or 3 reviewers, and either 1 or 2 of them were double blind, and one of them was so every single poster that was submitted to COSYNE was partly double blind, partly non-blind.
Right.
Did they talk about what they might do about this? I mean, it's interesting. It seems like it's sort of embarrassing.
Yeah, it's a little bit embarrassing, of course, but it's also really cool that they're transparent about that. I mean, you could see a lot of it mostly in the affirmative action taken on enforcing diversity. I'm not aware of, that they're thinking of doing anything drastic, but I just thought it was.
It seems like the conclusion is pretty obvious. You should just always do double-blind reviews.
Yeah.
There's no. What is more is there to discuss here?
Yeah.
The more experiments you do, you're actually hurting the people who are not double-blinded.
Yeah. Well, right? Yeah, that's true. Yeah.
Well, what about open review? Did they discuss this at all?
No, they did not discuss that, and I think it's a fascinating thing. I'm a big fan of open reviews. Talked with. We don't really have time to go through all of them in detail, but I just kind of want to browse through at least a couple of them to give you some sense of what I've been looking at. So, for example, there was this interesting paper from PIU, like a student at Carnegie Mellon, where they showed that there is, in fact, microstructure in mouse V1. It's just that the cell clusters are tiny. So, like, these clusters are at the most 20 microns. So we're talking about orientation columns in mouse V1, where people have argued for a long time, look, it's just salt and pepper. There's no organization whatsoever. That is not true. They've now shown pretty conclusively with 2-photon calcium imaging work that there is some. It's just that it might just be 3 or 4 cells that have the same orientation tuning, immediately next to each other. It might not be like a big, like a big ass column as we see in higher mammals, for example.
So this, I want to make sure I understand this, Florian.
Mm-hmm.
This is sort of finding equivalent to mini columns. Is that what this is?
Yeah, you can argue about it that way, because they're trying to explain the data with a layer 4 to 2-3 projection and some very regional recurrent connectivity. So I guess at least for the superior layers, it's an argument for sort of embedded, minimal functional columns. Yeah.
So you're saying the minimal, first of all, you said they're about 20 microns, so it's just very small, which would make them hard to see.
Yeah, they're really hard to pick up, but with 2-photon calcium, you can prove that they do exist.
So what makes them more minimal than another mammal? Other than being very, very skinny, but that doesn't really change necessarily the functionality of it.
Right. So I was trying to ask them, well, how deep could you go with the 2-photon calcium? But as far as I understood, they really took only like layer 2-3 into account here. And then it tends to be like, I looked at the raw plots on the poster, and it's like 3 or 4 cells that have the, same tuning, but they're right next to each other. And you see that in a couple of different places. So there's definitely like some grouping of orientation selectivity, and it's most likely because of shared inputs.
Yeah. This is a very important result. I'm glad you brought it up.
Yeah, I thought this was important.
Yeah. So it's important for 2 reasons. One, I've always believed there has to be something like mini columns, even if they're not visible, they have to be functionally there, and it just didn't make sense otherwise. And so this is good data for that. And then the second thing is just to realize how small they are. So in their area, if it's really a 20 micron spacing, that's important, because typically in a mouse or rat, we think about the whisker column. It's about half a millimeter in diameter, and if you're going to need a bunch of mini columns inside of it, well, if you're thinking about a mini column as 80 microns, you just don't get many in there. But here you can say, oh, okay, well, if I've got half a millimeter by a half millimeter and they're 20 microns wide, then I can get a bunch in there. And so it sort of gives you the numerical abilities to do some interesting stuff. So thank you for pointing that out.
Yeah, that's exactly why I thought it was important to us who focus a lot on columns. Next one was a paper by Josue Orellana. I'm gonna butcher all kinds of different names. I mostly took it down because I used in my own model a phase locking value, so like PLV metric, to capture phase relationships between my prefrontal cortex simulation and my inferior temporal cortex simulation. And it turns out that there's some cool multivariate alternatives to that, which he developed. He calls them torus graphs. And they actually used an L. Miller data set, which kind of had me listening to it. It probably doesn't mean much to you guys, but to me it was, oh, this is a cool analytical tool I want to show to some colleagues of mine. This was kind of cool. Dan Levenstein, I think, is that NYU? Yeah, that's Buzaki Lab. Actually, like rather simple kind of an analysis where you just look with sort of from the single neuron points of view, you look at interspike intervals versus firing rate coefficients. And to confirm what many modelers have, like, for a long time said, that when you look at the distribution of interspike intervals, that tail is actually way too fat. In the sense that when you plug it on a log scale, you see this bimodal distribution between cells that you might call in a ground state, so low asynchronous irregular activity, and cells that are sort of in an activated state. And then you can look at different brain regions and see that indeed there are like these, like you can see like these population clusters here, right? How there's like separable populations between like the fast spiking, less variable, and the regular low spiking. So this is just a very broad average, but you can also see that in some areas, like back here, like in CA1, you can see there's actually 3 kinds of activity in CA1. You have like spontaneous activity with a high coefficient of variation and low spiking rates, right? So this would be 1 Hertz or slower. Then you have this band here, which is obviously theta, right? So this is like 125 milliseconds or something, like this long band. And that is quite separate from gamma activity, this one here. And so I thought it was kind of cool to see in like amassed single cell like recordings here that you would get like these clearly separable populations that don't show up if you just go after ISIs or CVs. But if you plot them like this, you can sort of prove that there's this dichotomy, right, between ground states and activated states, and that different brain areas have like different repertoires of that kind. I thought that was kind of cool.
I have a question about that. It was interesting.
Yeah.
Yeah. One thing that's always puzzled me is sort of this, the fat tail there, the low frequency sort of background firing rate. And what does it mean? I mean, we can imagine assigning value to the gamma frequency or the whatever. But what is these sort of uncorrelated background rates? Is anyone talking about that? What those things are? Those things doing anything, or is that just what neurons do when they're sitting around?
Yeah, it's really interesting. I mean, in most of the modeling attempts that I've always seen, the background is sort of treated as just that, right? It's like a noisy background, and it's separate from an activated state. But it is quite interesting that in prefrontal cortex, for example, experimentalists like Earl Miller distinguish different kinds of activated states. So they're saying that, look, there's a lot of information coded in these gamma bursts, and that those are different from this beta activity, which is not background, but it's a separate frequency that is, and you get like when monkeys are like essentially allowed to forget stuff, let's say at the end of the trial, you get like these flushes of beta activity, which is sort of like almost like a memory wiping signal or something, if you wanted to think about it in normative terms. And so that doesn't explain, what the ground state is doing, but at least it's arguing that sort of there are different roles to different rates and coefficients of variations. I know that doesn't really exactly answer your questions, but what I'm saying is some people are thinking about what different states. Right,
yeah, I think that's great. So you can see these gamma and beta states. You can really believe that. And the only question is anything interesting or anything meaningful happening during these, background states? I don't know. I just think that maybe you were talking to these people. Maybe they said, "Yeah, we think this is what's going on." If they didn't say that, then they didn't say that.
Yeah. No, I mean, they weren't making any strong claim here. They were mostly sort of like pointing out that, look, wherever you look in the brain, there's like these separable populations if you plot them like this, right? And so we better think about why that is and have a good understanding what circuit mechanisms that are and what functionally they do. Anyways, I think we should move on. So next one we have David Tingley, also at Buzaki Lab, which was really cool. I never heard about lateral septum neurons, which are actually like downstream of hippocampus. So if you look at hippocampal efferents here, you get like inputs from CA1 and CA3 in these neurons, and these neurons are very peculiar because they have like different modes. And one of the modes of firing that they have is that they're like phase processing over the entire freaking arena. So you have like these D-shaped running tracks or circular running tracks. And what you essentially see is if you see the color scale here, right, on the CA1 theta, so you have a late one in blue and middle one in yellow, and an early one, middle one in light blue and early one in yellow. You see like the theta when the rodent is like, I think they are mice, when they're running around, the theta phase is processing over the entire fucking arena all the way to the start. So it's kind of a little bit nuts that if you look then at like rate correlations and phase correlations, you can decode the position like super tightly just by the phase of the spike, right? The theta phase of the spike, rather than, you know, what we are thinking of a lot in terms of like place cells that, well, okay, you can read it out from the firing rate of specific neurons that are specific to a place. No. This neuron is specific to a place, but it's specific with respect to the phase. It's actually firing everywhere. It's just that it's firing in different parts of the theta cycle, depending on where the rodent is. So like what kind of baffled me was the scale of the theta precession here. Those are like downstream neurons, so that's lateral septum, and they're like, he was talking a little bit about the fact that these neurons seem to switch between different modes. So in one mode, they are essentially transforming these the sequence code, right? Like doing replay, doing sleep sharp wave ripples, into a rate-independent phase code for spatial position. So this is like this whole theta over the entire D-shaped arena, which I thought was a bit nuts, but that's really cool to see. And then in a different mode, the actual content of the compressed sequence is completely filtered out, so you don't know what sequence is playing, but instead you get a rate code for the magnitude of the preplay or replay event in CA3, CA1 that you see there. And so he was talking about like the switching dynamics, and they have like an early plastic model to try to explain how these neurons in the lateral septum come about and why how can they have coding like this. And so I'm not claiming to like fully understand this. I think I actually want to read the paper, but I was just mind blown by seeing theta precession on that scale.
You know, what's interesting about this, it seems to me that this has to be learned for the environment. That
yes.
Otherwise the animal wouldn't know where it is. These cells imply the animal knows where it is on this circuit, and you wouldn't know that until you've learned the circuit. Where theta precession, as we see it in place cells and grid cells, does not need to be learned per environment. That's just an inherent property. So that's right. A very interesting distinction that it's a bit puzzling.
Yeah, which is why I thought it was cool that they're actually building a plastic model that explains the emergence of this through like exposure to repeated replay sequences, like long sharp wave ripples of place cells that run through the circle or the D in this case. And to how do the downstream neurons, right, learn to build these different modes, right? Yeah, I thought that was totally cool, and I intend to read on that.
Hey, a quick clarification thing. When you say omitting sequence content, what exactly does that mean? What are they omitting here?
Right. Like apparently you cannot like read out the specific place cell sequence that played, but you instead read out very tightly its magnitude. So it's, you know, like the amount of activity.
Okay, so you couldn't tell whether it's going clockwise or counterclockwise.
For example, yeah,
track,
right. I mean, it's a linear track, so there's not that many possibilities.
Mm-hmm.
But yeah, that's what they were saying. So yeah, next up we have, what was his name? Thibaut Terfumier, which used to work with Eli, I guess, when he was in Austin. So I was just trying to count the different ways that place cells can put their fields. The argument being that if place cells are built downstream of grid cells, which is of course a part of much of Elafit's thinking, then you're not actually free to place your place cells where you want. And so there might be many different ways to arrange them based on the input coding range, but they're essentially arguing on the basis of mathematical modeling that the number of place cells that are actually realizable, or like the arrangements of place fields that are realizable for a place cell, make up a vanishing fraction of the potential arrangements. And that there is a benefit to that, though, namely that you get larger separating margins between place cells, which is kind of like a stability for the price of capacity. There was more on this in the talk that Ela gave, where I essentially pointed out that, of course, there's this interesting behavioral timescale plasticity that we've seen by, I talked about the work of Aaron Milstein, right, who's now at Stanford, who's actually also at the meeting, where they could put place cells kind of where they wanted to, but within like a rather broad temporal range. So they would do like this dendritic plateau potential. They would, activate it at a certain time, and then you would pretty reliably get a place field in a window of like, -3 to +2 seconds on that track from where you stimulate it. So that kind of like suggesting that, well, you can decide to have a place field, but we cannot exactly tell you where it's going to go. It's going to go somewhere in this range here. If it's possible at all, it's going to pop up, in this neighborhood. And so it might be an interesting question to ask about the architectural constraints, like in terms of how many grid cell, inputs could you possibly have on a place cell that could then be associated with this dendritic plateau potential to build a place cell where you want it to. And in fact, there were a lot more talks about this whole topic of this interaction between grid cells and place cells. I'll talk about that in the recap of the grid cell meeting. Then we had this poster here, Alex Hrubinsky at Purdue, which was kind of interesting because we were talking about theta and V1. And so they analyzed, this is in vivo whole cell patch clamp recordings of visual response in awake mice. And they showed that the direction selectivity was much improved with the emergence of theta oscillations. And so I didn't fully understand all of this, but there's a conclusion that we might want to read. What was it here? It was, I put it in here. So they found that familiar visual stimuli could induce theta oscillations in V1. So there was like a training effect to it. The membrane oscillation was especially prominent. While theta oscillation was induced, the visual response firing rate of neurons was decreased after familiarization. The direction tuning recording showed the response of the neurons to their preferred orientation was relatively unchanged, while the response to the opposite direction was significantly decreased. So you gain, right? Yeah, this led to increased direction selectivity in layer 2-3 neurons. And then it makes some argument about like changes in the like layer 5 projections that I didn't fully understand. Apparently there's some computational model that I might want to take a look at. I mostly thought that might be interesting to take a look at this in the context of the standing and traveling waves paper that me and Jeff have been reading. Then next up, we have David Turrell at MIT. So that's Wilson Lab, who's been looking at what do these different kinds of replay do. So arguing that the hippocampus has two different states. One is the theta state, in which case you get like these processing awake replay sequences, which are sort of like short and sweep ahead of the animal once per theta oscillation. And then you have this much more massive shortwave ripple, a sleep time reactivation. And so trying to map that onto what you might possibly do with these things. in terms of hierarchical planning scheme. So if you're navigating an environment, I don't know, and you're a rodent, you're right here, and you want to get back there. Obviously, in robotics, I mean, people have been building like these learning rules, right, to learn like gradients to go to the target. How would the brain possibly implement that? Is sort of, you know, hard to say, because you don't get like full replay to the goal target. But what you might instead get is like sort of like a partial hierarchical planning scheme, where the prefrontal cortex computes sort of like an intermittent goal, and then that intermittent goal is actually in the reach of the theta sequence. And the way that the PFC knows where to set this intermittent goal is the much longer sharp wave ripples during sleep, which can connect much larger areas. And then, so if you're planning a path, you can have like, I analogized like this map a little bit. So they have like a cost function here, right, on this floor that like needs to be navigated towards some target. That it's sort of a little bit like there's puddles, you know, or like, you know, sand dunes or something on the way. And so if you're planning your way, you kind of want to like dodge around them. And they essentially like link up ideas of this, you know, this hierarchical planning, this efficient trajectory for flexible. Us about the evidence between like how place cells and grid cells interact, particularly how grid cells compose possible place cells, which ties in with the other student of Ila's that I was talking about before.
Florian, do you think that was a robust result, that place cells that had multiple firing fields in one environment would have multiple firing fields in another, and place cells that don't? You think? Did you believe the experimental results in that case?
Yeah, I saw the number of like recorded cells that they had with plotted over like the number of fields, and I think it's believable. Yeah.
Is it possible that they? I mean, the sense that like grid cells have different spacings, and you know the different modules are spaced out sort of at 1.4 times the next one. Could it be related to that? Could they be looking across different essentially different modeling systems, like a place cell that's associated with grid cells at one scale would naturally have more firing fields than a place cell associated with grid cells at a different scale, and that would be a very simple explanation for that.
Yeah, I don't think this has much to do with the scale of the environment. I will return to that, because they actually sort of try to. This researcher at NTNU, Cliff Kentros, who is like actually like trying to meddle with all of that by meddling with the grid cell activity and then seeing what kind of remapping he gets with in the place cell realm. And so I think there's more to talk about. But the finding that somehow there might be like a hidden attractor landscape of sorts that makes some cells more likely to have firing fields. And that stays over across environments, because some of the firing correlations between place cells are also like preserved.
Well, we can come back to it later.
Yeah, we can.
It seems to me there's a very simple explanation for this, if you, but you could miss it if you weren't thinking about it correctly. But I'll let it go.
Right. Then this one ties in with our whole discussion of the Behrens lab, or like the Constantinescu paper, I should rather say, right? About hexagonal directional coding. So Seong-Min Park at UC Davis did a study like that. I don't know if you've seen it already. I think there's a preprint out, in which they trained people in. So let me see if I get this right. So they had like these people, like they had faces, and they assigned them like two different values. I think it was something like you know, what were the two dimensions? Something like yeah, popularity and competence, exactly. And so, but people didn't learn like this whole map of all the people and where they're at the map. What people learned instead were like directional rank comparisons, where they would essentially learn that, you know, one person is more competent than another person. And this person is, you know, more popular than that person. And like you learn all these relationships. So they never get to see this map, but they're kind of like intrinsically building it, because the task that they have to do is they have to essentially like select business partners, and you kind of need people that are like popular and somebody that's competent. So ideally you need to sort of be able to compute like the spanning vector of these things to get the value of a pairing, because it's no good to compare somebody competent with somebody that is, you know, almost equally competent. You'd rather, you know, pick somebody that is like very popular and then probably less competent. And so the cool thing was that in training this task, people had to navigate this made-up space with faces that are assigned sort of this metric of sorts. And so very similar to, you know, the stuff that in Constantinescu, where people were navigating this neck and leg space, people are now navigating this competence and popularity space, except with people's faces instead of, you know, bird pictures, and assigned competencies instead of like these, assigned faces instead of like, you know, symbols. And then we're looking for the hexagonal symmetry, you know, using an unbiased way. So like refining a little bit the stuff that Constantinescu was doing to show that, yes, indeed, there is hexagonal symmetry of some sort. It's provable. And it's not just entorhinal cortex. It's also medial prefrontal cortex and the temporal-parietal junction. And I don't know what STS is. Did a couple of like cross validations. And then they did what was like one of my first questions when I saw the Constantinescu thing, is like, you know, what is the actual angle of their grid? And the cool thing is that they could at least actually answer that, because there's like a bias in some of these things, and they could try to link the directionality of the grid in entorhinal cortex to the directionality. You see like these links here between the directionality in entorhinal cortex with the directionality in dorsomedial prefrontal cortex, where they found, you know, strong signatures of this hexadirectional tuning. And indeed, they showed that these angles are kind of like the same. So they have the same bias towards a specific angle in that map that you are spanning out, which the people never saw but had to build for themselves, and which has, you know, like apparently these are correlated. What I was, of course, like trying to get to, well, is this angle driven by entorhinal cortex? Like, do we use the spatial mapping system first to then entrain sort of hexadirectional tuning in other areas? I don't think I got like a clear answer to that, but it's very interesting. They're looking at additional areas now, and they have you know, like slightly more complex tasks, and they're essentially showing the same stuff again. Proving that that original Behrens paper was, you know, obviously, a new way to think about how we are coding like these cognitive tasks.
So
we must have seen this paper or some other similar paper, because I remember this task.
A year ago at CNS, probably minutes before I met Florian, I went to this same poster. This person was presenting an earlier version of it. It may have literally been the same session that Florian was presenting a poster. So Yeah, so this one, what is really significant about this to me is that the person is, you use the word navigating a few times, but like the person is not, there's no control stick. A person isn't moving through this space at any point in time. There's nothing spatial about this.
Right.
There's nothing, the person is not moving. They're not path integrating. Where in the Barons, the Constantinescu one, the person is. And so it's really interesting that something like here, there's no continuous variable here. There's nothing continuous here. It's just like this, this person's greater than this person. It's just that. And the fact that the 60-degree thing replicates for that kind of space is totally new, if you can believe it. Like,
yeah, it's way more cognitive a task, right?
Yeah, and it's just not, yeah, sure. And it's just not like these continuous spatial variables that you're moving between smoothly. It's something different.
I was just thinking if I can like actually find the actual abstract here. Let me see, what was the number of the post? 2108. So that was the second poster session. That was on the Friday. 108, here it is. No, that's not it. Did I confuse the number? Oh, it's 107. Haha. That happens when you type your notes too quick. So, right, participants learned the status of people in independent two-dimensional social hierarchies, piecemeal from binary comparisons. The overall structure of social network was never shown to the participants. In fMRI, participants were asked to choose the better collaborator for an individual in the social network between two by comparing their growth potential, which corresponds to the area drawn by the selective rank of the two individuals in the 2D ability space. First, the dissimilarity in the pattern of HC and EC activity increased with pairwise Euclidean distance between individuals in the 2D social network, suggesting that social dimensions learned piecemeal are integrated into 2D cognitive map. Second, the EC, medial prefrontal cortex, so MPFC, tempoparietal junction, that's the TPJ, and posterior medial cortex. My God. All display hexadirectional signals for the direction of inferred trajectories over the cognitive map that guide decisions. The grid-like signal was specific for 6-fold periodicity and consistent across sessions more than a week apart. Yeah, so the multidimensional cognitive map. Our findings show that a grid code in the human brain is extended to encode inferred trajectories for decision-making over an abstract and discrete cognitive space, which suggests a general mechanisms for how the brain implements model-based decision-making. Ta-da. Yeah, so this was pretty cool. Couple more things. So I'm now looking at this paper. What's his name? Kohitishkar, like this MIT DiCarlo lab. We're just looking at the interaction between PFC and ITC during object recognition and like the role that recurrence plays, which was kind of interesting to me because that is exactly what I did during my working memory tasks, sort of trying to understand that, building that associative loop. And so I want to look at that and see what they say about the time delays of that recurrent integration, where essentially prefrontal cortex helps inferior temporal cortex resolve the object. So that's more stuff for me to read, as well as this one, which is a working memory for temporal estimates. So the question being that, I mean, we've long known that like people have been looking at discrete working memory tasks for a long time, like storing objects or, you know, colors or directions or, you know, saccade spaces, whatever. But it's rather unclear how working memory would work for continuous variables. In this case, that continuous variable being like a temporal delay. So they have a task where you need to like encode a delay. See if I can find the abstract on that. That's like 347. Let's see, that must have been the Saturday poster session. Yeah, here it is actually. So people have obviously proposed solutions like models for this, like line attractors. But if people theoretical considerations have questioned whether neural systems are able to establish robust and stable line attractors. And so they tested this with a continuous variable, namely time. So the monkeys are trained to measure a time interval drawn from a prior distribution, hold it in memory for a variable delay, and reproduce it afterwards. And then you can ask whether the noise that you're getting, the error that you're making, is explained by the variance that you get just from like the longer delay, or to what extent there's bias in the system, essentially like a systematic drift towards the mean value of the prior distribution of delays. So like if you no longer know exactly, because you kind of lost track, how long the delay is, you drift towards your mean. And they show that much better explains the data somehow. They even built like a recurrent neural network to show that, like I didn't fully understand it, but essentially you have a faster and a slower manifold, and you put the temporal encoding on the slow manifold. Let me see. So they had, looking at the dimensionality of the imputation of this task, analysis of dynamics revealed that the recurrent neural network relied on a hierarchy of slow manifolds to implement the task. In particular, the temporal interval was held in working memory using a dynamical landscape close to, but distinct from, a perfect line attractor. This deviation from a line attractor causes slow drifts of the representations in working memory, creating biases similar to the monkey behavior. So I thought it was just mostly like cool to see people like pay attention to the fact that working memory is a lot more than discrete items, and trying to build proper models for it. It still feels to me like it's early days for continuous working memory somehow. But what do I know? More papers to read. So just some of the posters that I looked at. Most importantly, obviously, I want to talk about the 15 years of GridCell, the workshop. As you can see, that is the list of the people who were speaking.
Hey, Florian.
Yeah.
So you said you're going to do 2 posters. You did all of them, I think.
Yeah.
And it's been an hour. So
right.
I'm wondering if, imagine that this GridCell thing is going to take a while.
Probably.
Yeah, and since we're trying to keep our research meetings to about an hour,
right,
it would be wise to do this maybe on Wednesday.
Yeah, I mean, like, sure. I don't know
if other people want to go through it. I don't know, but I can imagine this will be at least another hour.
Yeah, I mean, I'm happy to hear what people think, if they want to hear more. Or I mean, like, in any case, I know that there's like more to talk about than we have time for sure.
I mean, I want to hear this stuff, but I'm just afraid that if other people can speak up, but this will take us well past.
Well, I mean, it's better if it's interactive, I think, from our standpoint. Yeah, I don't want to short circuit the workshop discussions. So if you have a lot of stuff you want to go into that, and if you want to go into that in detail, then Wednesday might be a better time.
Right.
I mean, I'd rather spend 2 hours on Wednesday than feel like we're rushed to get it done in 45 minutes here or something like that.
Yeah, sure.
But that's just me. I mean, you guys are all working on stuff that's that's totally unrelated to this right now. So maybe Subutai and the rest of the team can speak up.
And were you going to talk about any of the oral presentations as well, or not?
Yeah, by oral presentations, you mean the actual talks? Yeah, for sure. I mean, I have notes from all of the talks, and we probably want to be a bit selective about that too. But yeah, sure. I mean, I want to be like interactive about this too. Like, you can tell me if if like certain pieces are not like immediately relevant, or if like some things you want to know more about. I mean, I'm happy to just provide like a very coarse overview. But of course, I have some 30 pages or something of notes for these different talks. And so there's quite a lot to talk about.
With the posters and stuff, I think you focused primarily on experimental data.
Mm-hmm.
Were there any interesting models that you saw or anything like that?
Yeah, a couple of different ones. I think that, most of the workshop talk to me was like rather experimental. But of course, it always has like model implications, and there were a couple of modelers there. So not all of it is experimental tasks. Also, like people like Menovita were just, like being very rigorous about what do we actually now know about the microcircuits that we didn't know before, which I think is also interesting from a model building perspective. But also, yeah, just very experimental stuff like, Ulanovsky, letting bats fly. And I think the more interesting stuff that maybe worth talking about is also like all the discussions that we had, like group discussions about what does the field think that grid cells actually do. So it was quite a mix of different things, really. I don't know what is the best way to sort of to like let you guys, like help guide this, right? Because I'm obviously happy to talk about any and all of it.
Yeah, I was just curious about modeling. And did you see any models of cortical columns or, what different layers in the neocortex might be doing or anything like that? There's usually a little bit of work on that, even though, not a lot typically.
Yeah, I must say, was very little of that. A lot of talk about like potential interactions between grid cells and place cells and downstream effects, and how we think about head directional coding, and people like Ilafit thinking a lot about sort of principled capacity of place cell coding and whatnot. But no, very little about columnar models in general. So I wouldn't say that people went that way. No, it was way more experimental than that.
Well, the problem is I'm interested in all this stuff, and so I'm almost, in one sense, I'm interested in. I've got some things I want to do today as well, but also I don't want to shortchange these things, so it's sort of torn a bit. I desperately want to know what people think grid cells are doing, how they think the interaction between grid cells and place cells and head cell direction cells. That's what I'm working on right now, and I just don't know how do we parcellate it out so that we have enough time for it. If there was a 15-minute topic, I would do that. But I think we went through all the stuff you've got. It's going to take hours. So I don't know. I gotta have to look for you for suggestions, Florian.
Right.
You tend to talk longer than you think you're going to talk, which is okay. I'm not complaining about it. But you say, right. I'll just give you a couple of these posts. Well, we do them all, which is good. I like them all. So I don't know. If there's a succinct topic you want to do, or
all right. Maybe we just do like two more quick things, right? One is that exact discussion. I named it here Science by Vote. And the other one, what was really cool, I think, Caswell Berry scaling up environments dramatically for rodents, which I thought was also really cool. So like one experimental thing and one like overview of
why don't we start with one and see how long it takes?
Right. Okay. Cool. So let me start with like this group discussion. I think it came about because we actually lost a speaker in northern Italy or something, and so we suddenly had like a spot free, and so Neil volunteered to sort of lead a grid discussion, essentially just putting out the question. Well, we have a lot of like really smart PIs in the room. What do we think grid cells do? First one, path integration. 2, vector navigation. 3, statistical and structural learning. 4, planning, sort of grid cells as eigenvectors of the task state space. So very, abstract cognitive. Who here believes that? Are we missing anything? And like speak up if you want to argue for some of these things, right? And I was like joking a little bit with Eli about, well, this is how we find out the truth, right? We're just voting on it, which was, like tongue in cheek, right? Overall, it seemed like there was a lot of caution in the room, which is not surprising that given that the conference actually featured a lot of caveats with like Trevor's talk, for example, like the end of the age of innocence or something. So people pointing out in all the ways in which the things that we know are not ideal, and grid cells are not ideal, and there's non-canonical cells and whatnot. So, but just like a broad reach of hand was, I noted down the vote. So it was about half of the people that were confident to say that grid cells do path integration. It was way less saying that, like about half of that, so maybe a quarter of people who were convinced that grid cells do vector navigation.
Can we just stop right there for a second? I mean, those are surprisingly low numbers for me.
Yeah, that's what I meant by people were very cautious.
It just seems like, of course, that's what they do. I mean, I don't know. I'm just surprised that people wouldn't have like 95%.
Yeah, me too. But I think just there were like just preceding that discussion were a lot of talks with caveats and people pointing out that, well, we don't really have vector cells, and there's a lot we like still don't know. And so I guess people were just like being cautious and sort of like abstaining
I guess
from voting.
So. I mean, I think the data that says these cells do this is scarce, but the fact that we know the brain has to do these functions, and grid cells are the first thing we've ever heard of that could do these functions, and they seem very likely to do these functions. That says, well, then that's why the certainty should go up. It's not that there's proof. It's more like, yeah, of course, this is like something has to be done. We haven't had anything to do it. Now we have something to do it, and it looks like it. So that's my take on it.
One interesting point is that there is a sizable group of people who think 1 is true, but 2 is false. And I mean, that's interesting that there are theoretically minded people. Let's see. There are theoretically minded people who think 2 is false. I can tell you that confidently. Because grid cells.
Go ahead.
Oh, just because with the grid cell code, there's no real trick for reading out navigation vectors. You still have to do a big, essentially, lookup table between pairs of, between, like, for example, our displacement cells. If we have a group of displacement cells encoding a navigation vector, to be able to read that out, you still have to do essentially a lookup table. And so people are a little bit down about vector navigation.
It's interesting that you say that, Marcus, because that is exactly the argument that Mirko was making at the meeting. Like pointing out, look, I mean, it's all good, but like, I advise caution. Like this might work for like a short range stuff, sort of, like on the scale of a theta cycle, you might stitch together a vector or something, but you can't use that for big whole area navigation. You won't get vectors, like vector representations like that, because exactly you would need like a lookup table, right, to somehow stitch it together.
Yeah, so I mean, that to me says that we're not understanding it correctly. Another way you can interpret this is, do grid cells on their own do this? No, I don't think on their own they do it, but do they? Maybe that's what people are reacting to. But it seems to be they are sort of a essential foundation from which the mechanism would be built. So maybe that's a way people could disagree about this. I look at it as essential foundation, but maybe on their own they don't calculate this, but they're part of the solution. So that's a different take on it.
Well, I mean, like both Burak and Burgess actually argued for 1 and 2, but also said that, like, to say that with certainty, we need to understand the interaction with hippocampus a lot better, which we are getting to. I mean, a lot.
That's my point. Right? My point is that it's on their own; they don't do this, but they are part of the fundamental mechanism that does it.
Exactly. So the vote on whether the grid cells do statistical and structural learning was like a little bit less than half, so like 45% or so, which is still, a reasonable vote of confidence, given that it's, almost the same as the one for path integration. And then the vote on planning. So like Neil Burgess gave like a very quick talk about this idea of computing grid cells as eigenvectors of a state space. So essentially, like a model of place cells, and then you have we just put place cells in an environment, and then you have a simulated rodent that just like runs a trajectory through it and stitches together a state transition matrix, right? So I have like these activated place cells, and from there I can transition depending on how I'm running, right, into these, and these. So you build a big transition state space transition matrix, and if you compute eigenvectors of those, they, after a long time, they tend to look like grid cells. And so if you abstract that out, then you can make some argument that you can do planning like that. But I think the room was quite skeptical. Was like, even after the talk, was 20% or so of the room that said like, yep, I'm confident that is true.
What's the difference between statistical and structural learning? I mean,
to me, it's kind of like the same.
Okay, so when I think of structural learning, I think of the stuff we've written about, like, oh, I'm trying to learn the structure of some system or an object or some space of knowledge, and therefore I'm placing knowledge in a framework that allows us to navigate in it. So that's what I think of structural learning. I'm not sure what they think of it. I'm not sure if it's the same thing. So
maybe structure is a bit more hierarchical than just statistics.
And I don't know what statistical learning means. I have no idea what that means.
I mean, like there was an anatomist that spoke up in that discussion. I didn't like catch her name, but she pointed out that there's like lesion data to suggest that. But there was like immediately afterwards kind of like a bit of a discussion in like whether or not certain lesions impair capability for what they call structural learning. And the room seems like to agree that there's good studies to say that is true, but also that is not true.
Does anyone know what statistical learning is as a separate thing? I don't know what that means.
I mean, like I don't think I would want to like give a definition, at least not in sort of
Well, I fine, but if people are voting on it, someone must have had some idea what they're voting on. So. We can.
A lot of people think that just typical neural networks, artificial neural networks, and stuff are examples of statistical learning, where you're just learning about the statistics of some input space, and based on that, you make some inferences.
But wouldn't that include all learning? I mean, what would you learn that's not statistical?
Well, so the old style, AI style machine learning, where you learn rules or you learn, things like that. Those would not be considered statistical learning. But if you're just looking at it as numbers and try to figuring out what the space of the numbers looks like and how you might carve up the space into different categories, or making inferences or assigning probabilities to it, those kind of things would be, I think, more statistical learning.
So I think structure learning in this context is referring to, for example, one of the structures that we encounter quite often is 3D space. Another structure we encounter is the rules of a game, like a flashcards game or something like that. Like
right.
We have these various structures, like, oh, this is like a flashcard game. Oh, this resembles chess. Or like you have these general categories. This is 3D space. And I think that's roughly what they mean by structure learning.
So that would be consistent with my interpretation, I think.
Yeah.
But that would be different than statistical learning, I think, that Subotai just said.
Right. But it's a little bit less like the structure of a coffee cup. It's a little bit more the structure of like just a recurring thing that occurs across multiple objects or multiple tasks.
Yeah, but
like for example, like the relationships that people learned in the abstract cognitive task, right? The one with like the popularity and competence space pairing thing, right? Where you somehow need to build a map, right? Even though people might not think of it that way.
I guess I don't know why that's not like the coffee cup. To me, those are all the same. Like, well, of course, I've been arguing that. You've got some regularity in the world, and the only way you need to find a framework, literally a metric space in which to place the knowledge of that thing, and then it becomes actionable. So, and I think I answered I think going with Subotai's definition of statistical, I think I understand that.
So my hunch for jumping to number 4, I suspect it would, something I want to do, at least with my time, is spend some time understanding the use cases of an eigenbasis. What are the reasons encoding something using a set of eigenvectors is desirable? What does it get you? Like, there are some that it's like an ideal code for just representing stuff nicely, but like separating.
Like it's an invariant basis.
Yeah, but the second set of use cases I want to understand is how it enables planning, or how it enables you to predict things. Like I think that it's going to have I think it has a set of nice properties that I don't fully understand. I know some of the basics, and there's a reason why number 4 is on this list. There are some pretty smart people who are pushing.
Yeah, Surya Ganguly is thinking a lot about that, for example.
So it's funny to me, planning and vector navigation are the same thing. And you're basically saying, now that I've arranged some knowledge in a metric space, how do I get from one location to the other, or to a desired location? So whether, just moving my finger to a different point on the coffee cup, or if it's, or how to touch the power button on my phone, or, how to enact a law, and, or something like that, or pick the right partner. Those are basically the same thing. I'm with you. My memory about eigenvectors is very poor. I haven't really thought about eigenvectors since I was in college. And so I'd have to go back and try to understand it. Is that a fundamental thing? And if it was, I would argue if it really is a good way of looking at planning, then I would argue it's a good way of looking at vector navigation.
I think it's a little, I think proponents of number 4 are going to think number 2 is a little bit too rigid, that the brain is not doing like space. It's rather doing its sequences of actions, or it's figuring out like what is near me in a graph theory sense. I take this action, I wind up at this near node. I take this next action, I end up at this next one. It's somewhat spatial, but it's not spatial. It's not strictly spatial.
Yeah, well, I think, but in our world, or my world, is that the same mechanism could work for strictly spatial or not. It's really a column in the cortex doesn't know what it's modeling, has no idea what its inputs represent, and it just has to find regularity in it through a sensory motor interaction, and then it'll discover the inherent structure of that thing. Whether it's a 3-dimensional space, it'll discover that. If it's a 2-dimensional space, it'll discover that. And if it's some weirdo thing that represents some planning of high-level thoughts, it'll discover that. But that's the basic idea.
Although it's interesting to me that there seems to be like this bias towards 2D representations, which I think has something to do with a cortical sheet and the way that it sort of centers around inhibition works to bias us towards 2D grid cell modules. 2D lattice, when the task that you're doing, let's say it's cognitive, is a 1D task.
Yeah. So I was working on that this very morning. I want to get back to it shortly, which is, okay, so I really like this idea that the mini columns are representing 1D orientation vectors, if you will. And the brain, of course, has to discover 3D structure. We know it has to be able to do that. There's no way around it. And almost certainly it could do n-dimensional, whatever. So but then you have to project it onto this 2-dimensional array. And it appears that the inhibition is in 2 dimensions, and that's why you get this. So the question then becomes, like, what does that mean? How do we understand that? It may be the 2-dimensional appearance of grid cells is just a fluke because that's how the cortex tissue is organized, and it may not be the essential element at all. There's something else hidden in there because we really don't want to model things in 2D. But that's where the inhibition would force things to look like 2D.
Right. So, I mean, like my overall sense of that whole meeting in the end was that there seems to be like quite a bit of confidence that we're making progress faster than ever before, in part big actions and how that whole circuit works together functionally as well. But without any sort of strong, consensus. I think.
That's been like that for 40 years, right? So
yeah.
That's
Well, you have a bigger overview than me.
Yeah. Hey, I think that's a great. If you don't mind, I think that's a great time to stop here.
Yeah, totally.
And I would like to pick it up again. We can pick it up Wednesday. And if no one else objects to that, that's what I would recommend.
I'm perfectly happy with that, to take it to a Wednesday. Also, to all of you, right? I mean, like, if you have your favorite PI on this list, right, you can also just let me know, and I can, we can start with that. I'm happy to talk about all of these. I have extensive notes, so pick your favorites, right?
Well, if you get some picks, that's great. If not, pick your favorites.
Right.
And we'll just start going through them. And I wouldn't be surprised if we don't get through them on Wednesday. But
we'll see what we get, right?
We'll see what we get.
All right,
Florian.
Yeah.
Yeah. Can you share this?
I think you might. Yeah, exactly. Okay, cool.
All right. Thanks.
Hey, thanks, Florian.
Thanks for watching, everybody. I'm gonna stop.
Thanks, Florian.